Hello everyone, welcome back to a lecture series on overview on integration of cellular metabolism. We are continuing with heme metabolism. This is the third lecture where we will be studying the heme degradation, how heme is transport, and we will be also discussing the bilirubin metabolism. So, these are the concepts that we will be covering in today's class, how heme is degraded, how it leads to the formation of bile pigment, how bilirubin gets conjugated and circulated, what are the various tests by which we can detect bile pigment, and we will also briefly discuss the principle of phototherapy, right. So, fate of heme, right. So, hemoglobin is the major source of heme. We already know hemoglobin, when it is degraded, it has got us where hemoglobin is present, it is present in red blood cells, right, and red blood cells have got a lifespan of 120 days, after which they are degraded in the reticuloendothelial system, that is the splenic and hepatic sinusoids, and when they are destructed, heme is released. So, globin, a protein part, is released and heme is released, right. So, heme we already know during synthesis and the structure of heme, how it is a cyclical tetrapyrrole structure it is having, and these are the groups, right, methyl vinyl, methyl vinyl, methyl propionyl and propionyl methyl, right. That is a very important thing to remember, and I already told you that you should remember this structure and not the intermediates. Anyway, so we are discussing the steps of heme degradation to this. So, heme, free heme, right, once it has been out of the RBCs, it is acted upon by the enzyme heme oxygenase. Okay. Heme oxygenase, the ring opens, right, heme was a cyclical structure. So, the ring now opens to form a linear tetrapyrrole structure, which is biliverdin. So, there is a specific region where the chain needs to be opened up, and this reaction also produces carbon monoxide and iron is also given up. So, we already know that heme is actually a ferroprotoporphyrin. So, now again the iron is gone and it is now open structure biliverdin, right. So, what happens, this iron has got many fates, right, iron binds to ferritin, especially in this case. Carbon monoxide is actually a poison, it binds to hemoglobin, but the concentration is very low. Carbon monoxide has got a potential to inhibit electron transport chain complex 4. So, that is how it acts as a poison. Anyway, we got biliverdin. What happens in the next step? Biliverdin is acted upon, you can see this is the structure, okay, here. So, biliverdin is reduced by biliverdin reduction. So, basically this methynyl bridge, okay, is actually reduced to methane, methynyl is reduced to methane, this is CH2. Okay. So, this reduction of double bond happens with the help of reductase, which needs NADPH. Okay. So, heme to biliverdin by oxygenation and then reduction biliverdin to bilirubin. Okay. So, there is a characteristic color difference between these compounds. Mind it, this is cyclical compound heme, it is actually red or brownish in color when it is out of the RBCs. When it is transformed to biliverdin, it is green in color, and ultimately when it is acted upon by NADPH dependent biliverdin reductase, it is yellowish in color, yellowish orange, bilirubin, right. So, biliverdin and bilirubin are actually bile pigments. You should know that bilirubin specifically has got no function in body and it is excreted through bile, purely excretory product. Okay. You should have a quantitative idea. So, about 6 gram of hemoglobin is broken per day, from which 250 mg of bilirubin is formed, right. It is also formed in a very minute quantity, that is 50 mg compared to 250 milligram from myoglobin and other heme containing proteins, right. So, in total about 300 is produced and 80% is from the old RBCs that are destroyed. So, recirculation of that product, and 10% from ineffective erythropoiesis and rest 10% from degradation of other heme containing pigment and myoglobin, alright. So, this is basically the source or the calculation of total bilirubin that is present in our system every day, right. So, this is the roughly erythroid RBC source is 80% and non-erythroid are 20%. You should know what are these sub options, because these may be taken up and given to in a multiple choice question. So, one thing from here may be shifted here in an all except type of question to state the reasons of non-erythroid sources of bilirubin. So, be very careful, right. A very easy way to remember is, please remember all of these, then automatically something which is not these will be from erythroid sources, right. So, always be smart in learning. So, what is shunt bilirubin? Now, to study this bilirubin or metabolism basically, or how succinyl CoA and glycine is actually united to form heme, right, and ultimately heme is degraded to form bilirubin. So, what is shunt bilirubin? When a labeled glycine molecule, now glycine can be either labeled at the carbon or nitrogen. So, when it is labeled the nitrogen, it is either labeled by 15 N15 or carbon by C14. So, radio labeled or radioactive glycine is injected so that it is incorporated into RBC, and naturally after 120 days or 100 to 120 days, roughly varies around 3 months, the radioactive bilirubin will be excreted. This is normal. However, it has been found that around 15% of radioactive bilirubin is excreted in about 10 days. Why that happens? It is because it is this fraction is actually known as shunt bilirubin, right. That is bilirubin that is already excreted without being incorporated into the RBC. So, this is actually happening due to ineffective erythropoiesis, that is RBC formation or heme formation is defective. And this, can you name some diseases where heme formation is defective? Porphyria. So, especially in erythropoietic varieties of porphyria, the shunt bilirubin is increased, alright. So, basically shunt bilirubin is that fraction of bilirubin which is formation of bilirubin in bone marrow without being incorporated into hemoglobin, right. So, you should be very clear, right. This is the 10 to 15% of bilirubin, right. So, this is a very important short note MCQ or a Viva question depending on what type of exam you are facing. Now, let us discuss the structure of bilirubin. So, we already know that after bilirubin has been reduced, this is the central methane bridge that is connecting the two dipyrrole rings. There is no confusion in that, right. However, as we see that in spite of having multiple polar groups which are shown in red, bilirubin, free bilirubin is insoluble in water. Why it happens? It is because there is an intramolecular rearrangement that leads to formation. You can see this double bond is actually getting like this, right, and it orients itself so that there is a formation of multiple internal hydrogen bonds, intramolecular hydrogen bonds, that leaves no free polar group to be dissolved in a polar solvent. Therefore, it is not soluble in water. Water is the major polar solvent that is present in our system, right. So, it is because of this reason that free bilirubin is insoluble, right, and the central methane bridge actually becomes buried, right. I hope you have understood. Please note, you do not need to remember all these structures, just for a conceptual understanding why free bilirubin is not soluble in water. You can simply answer that it due to presence of intramolecular hydrogen bonding. Number 1, the central methane bridge will be buried, there will be no free polar group to act with any polar solvent, right. So, this is basically the ridge tile structure of bilirubin, that is a named nomenclature where the central methane bridge is buried and there are multiple intramolecular hydrogen bonds in spite of presence of multiple polar groups, right. So, let us see what happens once bilirubin is formed, right. So, once bilirubin is formed, you can see heme to biliverdin into bilirubin, it is actually combining with albumin. So, this whole thing albumin bilirubin complex will be then transported to the liver. So, first step is, since bilirubin is not hydrophilic, it means it is lipophilic, right. Therefore, it is easily bound to albumin, right, plasma, which is the plasma protein, and one molecule of albumin can bind to 2 molecules of bilirubin, right. And in generally 100 ml of plasma can transport up to 25 mg of bilirubin. So, this is the phase where albumin is bound to bilirubin, right. So, after combination of combination with bilirubin, it is then transported to the liver, where albumin, you should know that this combination of albumin is actually loose, okay, it is not very tightly bound, right. So, whenever there is any need or whenever there is any excess of bilirubin, this albumin can actually easily dissociate from bilirubin, or whenever some drugs that facilitate this attachment on bilirubin can easily displace the albumin. So, drugs like aspirin, penicillin, etc., they can actually displace albumin and they can lead to formation of free bilirubin, which being lipophilic can easily cross the blood brain barrier and get deposited in the brain, leading to the formation, especially in babies, leading to the formation, newborn babies, leading to the formation of a disease that is known as carnic terrors, where the whole brain is actually coated by bilirubin, and that is one of the very important condition that is present in newborns. We will be again discussing it sooner, and it leads to multiple neurological symptoms, and it can also lead to coma and death of the baby. Anyways, so especially in newborns, these drugs administration should be taken care of. If due to some need, we need due to some requirement, clinical requirement, we need to inject these drugs, precaution should be taken, right. So, bilirubin albumin complex, it goes to the liver. After going to the liver, what happens? The albumin, so bilirubin is, albumin is actually then dissociated. After it goes to the liver, albumin is dissociated, and now we have free bilirubin, which is again insoluble. But when it enters the liver with the help of a transporter, the albumin, the bilirubin, sorry, will get conjugated with glucuronic acid, which actually makes it water soluble. So, let us see how it happens. It happens with the help of UDP-Glucoronosyltransferase or UDP-Glucoronyltransferase, it is one and the same, abbreviated as UGTs. Is generally a group of enzymes, it generally this enzyme, this group of enzyme, it has got multiple isoenzyme, multiple isoforms, which acts on multiple subjects to convert the substrates to their corresponding glucuronide form. The mechanism is common to all. So, UDP glucuronic acid is converted to UDP, uridine diphosphate. Again, where you have learned, you have read UDP donor, where in case of glycogen synthesis, UDP glucose was the glucose donor, right. So, here with the help of UDP-Glucoronyltransferase, UDP-Glucoronyltransferase, a substrate will be converted to glucuronide. Bilirubin, just replace substrate with bilirubin and you already got your reaction, that is bilirubin will be converted to bilirubin glucuronide. So, this UDP-gluconyl or glucoronyltransferases are present in endoplasmic reticulum, and they convert many internal and exogenous toxic substances to the non-toxic metabolite. This is a part of xenobiotic, right. It is how waste products are metabolized in our system. They are a family of enzymes that are concentrated in the liver, and the enzyme isoform that is specially conjugating bilirubin is UGT1A1. Okay. It is essential for conjugation of bilirubin and its eventual excretion in the system, because I told you bilirubin is an excretory product, it needs to be thrown out of the system or excreted, right. And inherited UGT1A1 deficiency will cause neonatal jaundice, jaundice of the newborns. So, what happens inside the cell? It is conjugated glucuronic acid to make it water soluble, and it is actually the first carbon of glucuronic acid that combines with the carboxyl carbon of the propionic acid side chain in the bilirubin molecule. Bilirubin molecule has got propionic side chain, and what happens? The first carbon of glucuronic acid is binding together. But how does it make it water soluble? There are actually two steps. First bilirubin becomes monoglucuronide, then diglucuronide, but let us see how it becomes water soluble. There were multiple, this is the insoluble photo of the bilirubin, right, insoluble image where the central methane bridge is buried, intramolecular hydrogen bond. Once this propionyl side chain actually combines with glucuronic acid by two sets of glucuronidation, the central methane bridge is actually exposed. This increases the reactivity, solubility, everything. So, this is why glucuronic acid or conjugated bilirubin, this is what? This is nothing but conjugated bilirubin, it is soluble. So, there occurs two subsequent conjugation, first it is converted to monoglucuronide and then to diglucuronide. In most cases the reaction is complete, therefore 80% are in diglucuronide form, whereas only 20% are in monoglucuronide form. There are certain drugs like primaquine, novobiocin, chloramphenicol, there are multiple antibiotics and anti-malarial as well as hormonal drug like pregnadiol that actually may interfere with this conjugation and that leads to jaundice. We will be discussing jaundice in detail in our upcoming next class, where we will be going through a plethora of reasons, right, and this is one reason. If there is a problem in conjugation, there will be jaundice. Okay. So, conjugation happens in the liver. So, there are some differences in free and conjugated bilirubin. Of course, solubility is in water is an issue, however, both are soluble in alcohol, mind it, right. Inorganic solvent, very important, organic solvent I mean. So, plasma level unconjugated bilirubin or free bilirubin, this is also known as unconjugated, is more compared to conjugated bilirubin. In bile only conjugated bilirubin is present, there is no unconjugated. So, whenever it has been fully conjugated, then only it is excreted into the bile and ultimately into the small intestine, and that is the pathway of circulation of bilirubin. In urine there is no free or unconjugated bilirubin, whereas conjugated bilirubin may sometimes be present, but it is normally absent. So, both are actually absent, but unconjugated is never present. There is no possibility of being present in urine because unconjugated bilirubin will not go actually. I will be discussing, we will be showing the pathway how bilirubin is coming out in urine actually. Next, when we are discussing with absorption from GIT, unconjugated bilirubin has been shown to be absorbed from the gut, okay, but not conjugated bilirubin. Unconjugated bilirubin being lipophilic and diffuse, and when we are discussing Vandenberg reaction, it gives indirect positive and conjugated gives direct positive. So, now what happens? The water soluble conjugated bilirubin is excreted into the bile by an active process, and this occurs against a concentration gradient, right. And this is the rate limiting step in the catabolism of heme, okay. And this step is actually induced and increased by phenobarbital. So, what is happening is the conjugated bilirubin is conjugated, it has transformed to diglucuronide form, it will be secreted in bile via the, and ultimately bile is a liquid liquid actually which is secreted in the gallbladder, which is present in the gallbladder, and via the bile duct it comes into the intestine via the second part of the duodenum, right. It reaches the small intestine, ultimately it goes into the large intestine, where inside the large intestine it is converted to a pigment that is known as urobilinogen, right. So, this urobilinogen, when it is formed, right, 20% of urobilinogen is actually reabsorbed from the intestine and returned to the liver via circulation, that is portal circulation. That is a different branch of circulation with blood vessels, that is totally different from the systemic circulation or systemic blood vessels that are supplying the gut, okay. So, portal circulation generally a circulation shunt between the intestine and the liver. We do not need to go into the details of the anatomy, but know this, some form or 1/5 of this urobilinogen is reabsorbed and again resecreted. So, this is the part where it is reabsorbed and it is going to resecreted. This back arrow, this is actually the enterohepatic circulation, right. And what happens to the other urobilinogen? It is excreted in the urine or kidney, it is excreted via the kidney. So, since urobilinogen is passed through blood, a small fraction is excreted in urine, all right. And this urobilinogen, right, that is present in urine, it is due to the fact that it is reabsorbed from the gut. Otherwise, once it is in the gut, it is destined to go into the feces, which happens for the majority because bilirubin is converted to pigments like urobilinogen and starcobilinogen that are abbreviated as UBG and SVG, right. Both are colorless compound to start with. So, bilirubin or the bile that is present in the gut, the conjugated bilirubin that is present in bile in the large intestine. So, from small intestine it goes to the large intestine, it is converted to urobilinogen and starcobilinogen that are colorless products, but they are oxidized by atmospheric oxygen or oxidation, right. Oxygen is present in there. In large gut there are air pockets, these are air filled organs, our intestines, right. So, ultimately they are converted to colored products, and these are, that is the reason that both urobilinogen and starcobilinogen actually impart our characteristic colors to urine and stool, right. Urine, generally urobilinogen is not that much colorful, right. So, it gives a tinge of straw colored. So, normally urine is very thinly colored. When there is excess water, we are drinking excess water, our urine is supposed to be colorless, but whenever we are having dehydration, that is when the pigments get concentrated and we get some dark colored urine, dark yellow colored or light yellow colored, right. But stool characteristic colored is imparted by starcobilinogen and it leads to the standard reddish brown color of the stool, right. So, this is the pathway where we can see the physiological route of bilirubin excretion, conversion excretion, and this is the simultaneous biochemical changes that are happening. So, bilirubin is transported to the liver with albumin, right, where in the liver it is actually getting converted to bilirubin diglucuronide. This is transported to the intestine, right, and intestine in as a conjugated bilirubin via bile, right, and where it is converted to urobilinogen. Thereafter, urobilinogen is converted to starcobilinogen and then starcobilin, which is excreted in the feces, okay, and some part of urobilinogen is also transported to the kidney and is excreted in the urine as urobilin, okay. So, remember this is the pathway of bilirubin excretion. Now, we are discussing test for bile pigments. So, specifically you need to remember name of the tests, what we use in order to detect each of these components of bilirubin metabolism. So, if we need to test bilirubin, it can be tested by Fouchet's test, Mellin's test, Vandenberg's test, okay, Vandenberg's test. The urobilinogen is detected by Ehrlich's test, same Ehrlich's aldehyde test that we discussed in our class of porphyria, that was detecting urobilinogen and porphobilinogen, right. The same test is used over here, that is it is mixed with aldehyde reagent to give a pink color. And urobilin, Schlesinger's test, okay, this is very important. This is specifically the urobilin and starcobilin, it gives a positive test. It should be noted that these tests are mutually exclusive, means Fouchet's test, Mellin test is positive for bilirubin and it is negative for bilinogen and urobilinogen, urobilin. Likewise, for Ehrlich's test and Schlesinger's test, these are also positive respectively for urobilinogen and urobilin, all right. So, among all these tests that we have discussed, we need to know about Vandenberg test, the theoretical aspect of it, because other tests will be taught to you, I mean you need to perform to detect either bile pigment or bile salt eventually in your practical examination. So, the theoretical aspect of Vandenberg's test is very important, which is actually very easy, that is in acidic medium bilirubin reacts with a diazo compound, the diazotized sulfonylic acid, to produce a purple colored azo pigment, right, and it is the conjugated variety which actually gives the reaction immediately, right, and hence this test is called direct reaction or direct positive, and hence conjugated bilirubin is also known as direct bilirubin. So, it is synonymous, the same thing. So, what actually happening is here, we already know conjugated bilirubin, the central methane bridge is actually exposed and accessible, and this is where the diazo compound reacts with, leads to the formation of purple color, whereas in case of unconjugated bilirubin, what happens, if we directly treat with the diazo reagent, there will be no reaction, whereas if we need to treat with alcohol, if we treat this unconjugated bilirubin or free bilirubin with alcohol, then alcohol will disrupt this intramolecular hydrogen bond, because it is a much stronger, it has got a much stronger affinity to form polar groups with these polar bonds, residues, and then the central methane bridge will be, the buried bridge will be exposed, and then it will give a positive reaction. So, unconjugated bilirubin gives indirect positive or indirect reaction, therefore, unconjugated bilirubin is also called indirect bilirubin, right. So, one thing you should know, that bile pigments, that is biliverdin and bilirubin, and bile salts are totally different. You have been taught how bile salts are actually produced from cholesterol, and how by conjugating with taurine and glycine they are forming taurocholic acid and glycocholic acid, they help in emulsification of fat. Those are totally different, right. They are detected by Hayes sulfur test. So, the test, bile salt and bile pigment, students have got a tendency to mix them up. They are not the same. Bile salts are useful beneficial compound, but bile pigments are actually useless excretory product. So, finally, why are we studying all this heme synthesis? We already know, due to diagnosis of porphyria. Why we are discussing heme metabolism and degradation of heme and excretion of heme and bilirubin? Because there is a condition where there is an excess amount of bilirubin in blood. So, we need to know the normal value. The normal value of unconjugated bilirubin, we already told you, 0.2 to 0.6 mg per dl, whereas conjugated is much lower, 0 to 0.2. It depends on textbook to textbook, but generally conjugated is much lower or even negligible in some cases, and unconjugated bilirubin is a bit more, but totally it is always less than 1. Okay? Whereas in a situation where it exceeds 1 mg per dl, this will be referred to as hyperbilirubinemia, means more bilirubin, right. When the value crosses 2 mg per dl, what happens? The bilirubin gets deposited in the white sclera and skin, and this is also known as jaundice, alright. And the icterus is actually the Greek term for jaundice, right. So, you can see in cases of hyperbilirubinemia jaundice, the sclera, that is the white part of the eye, as well as the nail bed, skin, etc., they get yellowishly discolored, and with more and more value of bilirubin, even the whole body may get yellowish discolored. But what about the value between 1 and 2? It is known as latent jaundice. Full fledged clinical jaundice more than 2 mg per dl. And lastly, phototherapy in some cases, especially in newborns, is used to treat jaundice, right. Since we are discussing structure of jaundice, let us discuss phototherapy. What happens? I already told you during discussion of Soret band, bilirubin can actually, the intermediates of heme synthesis, since due to presence of pyrrole rings, were actually absorbing various wavelengths and spectrum near blue light. Bilirubin also has got a tendency to absorb ultraviolet light in the spectrum between 460 to 490. But the beauty of it is, when it absorbs, bilirubin gets converted to a water soluble form, that is known as lumirubin, that can be easily excreted in via urine. So, this is again the insoluble bilirubin, right. When we are exposing it to UV light, there is intramolecular readjustment. This molecule is again becoming like this, and the carbon atom that is present over here, this actually creates a steric hindrance. Due to this steric hindrance, the intramolecular hydrogen bonds are disrupted, and this is all a transient phenomenon, and transiently the bilirubin becomes soluble, the polar groups become exposed and it is dissolved in water. So, lumirubin, in spite of being unconjugated, is dissoluble in water and it can be excreted via urine, and the solubility is increased. And this we can see is a phototherapy bed. This is a blue light, the newborn baby has been placed in the bed and his eyes have been covered because ultraviolet rays can damage the retina. So, prevent retinitis. This is a phototherapy bed. We will be again discussing jaundice in much details in the upcoming class, but you should know, if we are considering the colors, the beautiful colors of bilirubin metabolism, hemoglobin when released it is red and brown, biliverdin is green, bilirubin is yellow, urobilinogen actually gives yellow color to the urine, right. Stercobilinogen gives red brown color to the feces, and bilirubin yellow color normally does not give any color, but if it is raised due to some reason, that is either impaired liver function or block in excretion, it rises in blood leading to the hyperbilirubinemia and jaundice. So, to conclude in this class, we have discussed the pathway of degradation of heme, how bile pigments are formed, how bilirubin undergoes an antihepatic circulation. Shunt bilirubin, we have discussed the difference between conjugated and unconjugated bilirubin, what are the tests, Vandenberg test, what are the mechanism of action, and the structural changes of hemoglobin during conjugation and phototherapy. So, these are my references and I thank you all for your kind attention.